Hi, my name is Danny Smith, and I chose primary congenital hypothyroidism for my disorder on this project. Some background information on this disorder is that this disorder is an endocrine disorder, and it is characterized by an abnormally sized thyroid gland. A normal thyroid gland is a butterfly-shaped gland in the lower region of the neck, and it produces thyroid hormones. These hormones are rich in iodine, and they are necessary for healthy brain development, growth, as well as metabolism in newborns. A shortage in thyroid hormone production is what causes the disorder, and this shortage of thyroid hormones is caused by an irregularly sized thyroid gland. Smaller thyroid gland results in lower production of thyroid hormones. There is one more cause of a lower production of thyroid hormones, and this is much less common, but this is in the synthesis of the hormone itself. A person might have a normal size thyroid gland, but produce lower levels of thyroid hormone because there's some kind of chemical problem within the synthesis of that hormone. As for the history, I looked and I looked and I looked, and I could not find any information as to when this disorder was first discovered or reported. So unfortunately, I have no information to report on that. So prevalence in the United States is that it affects 1 in 2,000 to 1 in 4,000 births. There is a larger prevalence known in less developed countries, however, because primarily due to the lack of newborn screening tests. Since newborn screening tests are what initially discovers that disorder in infants, not having that screening test available results in a higher prevalence in some countries. One more point to make is that it affects twice as many females as it does males. And according to the NCBI website, the reason behind this is unknown. Physical effects. Symptoms vary between individuals. Some infants might be sleepy, they might sleep more, be less active. Some infants might have difficulty feeding. Some infants might experience constipation, but a lot of infants show no symptoms at all. And in fact, most babies have no physical signs of the disorder, which is why newborn screening is so important. If untreated, hypothyroidism is known to cause slower growth of an individual, as well as potentially the most severe is intellectual disability. If treated within 2 weeks of birth, however, newborns usually develop completely normally and they usually have no symptoms. Hypothyroidism results from one of many possible gene mutations. However, hypothyroidism is largely sporadic, which means it's not always inherited. In fact, only 2 to 5% of cases are inherited, and in these inheritance types of scenarios, they are usually associated with the paired box 8 gene. And this paired box 8 gene is located on chromosome 15, and its specific locus is 15q 25.3 through 26.1. But I do want to mention, though, that these other genes here, actually TSHR and DUOX2, these are 2 other genes associated with the inheritance patterns, but these are very, very uncommon, so I won't discuss them very much in this presentation. But any one of these genes, if there's one mutation in any one of these genes, this could result in hypothyroidism because all of these listed genes are involved in the thyroid hormone production. Mode of inheritance. This disorder is actually, it's been observed to be both autosomal recessive and autosomal dominant inheritance patterns. However, the most common, I think I may have mentioned earlier, is autosomal recessive. But I will briefly mention that for the dominant form, this results from mutations in the TSHR, which stands for thyroid synthesis hormone, or the DUOX2 gene. And of course, with dominant disorders, it only takes one dominant gene to cause the disorder. However, with this particular disorder, the autosomal recessive inheritance pattern is the most common. and this is usually associated with the PAX8 gene, as previously mentioned. And for recessive disorders, in contrast to dominant, they require two copies or two recessive copies of the mutated gene in each cell for the disorder to show up in their genotype. Again, I'm going to use this as a template for further discussion in this presentation. It only takes one parent to receive this disorder. However, depending on the status of a second parent, whether they're a carrier or whether they are afflicted, this determines the probability for which the offspring receive the disorder. This disorder can also occur from mutations in formation of sex cells, and in this scenario, this could happen regardless of family history. So there may or may not be a family history of the disorder in this scenario. So there are 4 possible inheritance patterns for which we went through in one of the project templates, which I'll go through quickly. So scenario 1, both parents are afflicted with the disorder. I use T for thyroid. You could use any letter, of course, but I use T. In this case, both parents are afflicted, so both are homozygous recessive, and so all of the offspring will also be homozygous recessive, and so 100% of offspring will be afflicted with this disorder. In scenario 2, there's one afflicted parent and one unafflicted parent, and they are also not a carrier. So this means that they must be homozygous dominant. So when we run this through the Punnett square, we'll see that none of the children are afflicted, but they are all carriers. Scenario 3, there's one afflicted parent and there is one unafflicted parent, but they are a carrier. So in this case, each parent has at least one recessive allele, and so all of the offspring will have at least one recessive allele. We'll see actually that there's a 50% probability of offspring being afflicted, and there's a 50% probability that the offspring are carriers, but they are unafflicted, seen here. In scenario 4, neither parent is afflicted nor a carrier, so none of the offspring will be afflicted or carriers in this case because there are no recessive alleles. So this is the pedigree I made for one of the project templates, and all I want to point out here is that a recessive disorder, autosomal recessive disorder, can skip generations. So for example, Penelope and Dimitri here that I made up, they are heterozygous, so they don't have the disorder since they don't have 2 alleles. But since they are both carriers for the disorder, one of their children has the disorder, so it can skip generations. That's all I wanted to point out there. The gene responsible for the disorder is the paired box gene 8. This codes for a nuclear protein involved in follicular thyroid cell development, and this functions in expression of specific thyroid genes. So PAX8 also activates genes during embryonic development. And one more thing to mention is that it's not only affects the thyroid gland, but it also affects the kidney formation. So if there is a mutation in this gene, it could potentially affect the kidney in addition to the thyroid, or it could affect the kidney and not affect the thyroid. So this is just the nucleotide coding sequence for a normal PAX8 protein. All I want to mention here is that the protein itself is 1,451 base pairs long. However, not all of that codes for the protein. The portion coding for the protein is right under 1,200 base pairs long. And here's the accession number used to find that. And this is the amino acid sequence. So an amino acid, as we know, is the result of 3 nucleotides in a sequence or triplet. And so there are 398 amino acids, and this is taken by taking the coding section of the nucleotide sequence and dividing it by 3. Here's the accession number for that, and this is the sequence, the amino acid sequence. There are more than 15 mutations associated with the PAX8 gene for which causes hypothyroidism. All of these mutations are usually or usually caused by a frame shift mutation by one nucleotide or more than one nucleotide. Although the NCBI website mentions that there are more than 15 mutations, they only provided 3 specific isoforms. So these are PAX8C, D, and E. All of these variants result in frame shift mutations, but more severe than that is that they result in an early stop codon, which this explains why the result of this disorder, the distorter is a result of a smaller thyroid gland, or it misses the thyroid gland altogether. It's because there's a stop codon earlier on in the DNA sequence. So a normal PAX8 protein helps in regulation for development of a baby, of the thyroid within the baby. So a mutated PAX8 protein will not regulate the genes needed for that development, or not regulate it to the extent that they need to. Some mutations cause abnormally small proteins, and this is a consequence of disrupted protein production. The result of this is a smaller thyroid gland and less hormones, as previously discussed. Here are a few different types of the disorder associated with that. But the most common is this thyroid dysgenesis. This is the most common type of congenital hypothyroidism, and this encompasses all cases related to problems within the development of the thyroid gland, and this is again due to mutations. So there are some available options for testing, but really it just boils down to newborn screenings. There are 2 types of newborn screenings, and this is the blood spot thyroid stimulating hormone, or TSH test. This particular one is the most cost efficient, and it's more specific in diagnosis of the disorder. The second test is the thyroxine, or T4 test. This is more beneficial in detection of a rare form of hypothyroidism, which I'm not going to pronounce because I will mess it up. But it's not as beneficial in detecting premature infants that have false positive, or it's not as specific with the high frequency of false positives in premature infants. So some facts on this is that newborn screening of hypothyroidism is mandatory by government for most, if not all, states. The textbook actually says it's all states that require this. This is one of 4 most recommended conditions to screen for. One of the other ones is PKU. Another one is cystic fibrosis, and the other one is sickle cell disease. So that's just a fun fact, a bonus of the presentation. Most screenings are performed 24 hours after a baby is born, and this is to allow time to pass for neonatal TSH surge. And this is done because it reduces the risk for a false positive. Diagnosed infants need to be reevaluated after 3 years of birth, because it takes time for the thyroid gland to develop, and so they may not end up having the disorder. After they're treated for 3 years, their thyroid gland may function normally. But early detection is extremely important, and this is because a lack of treatment can cause mental retardation, and also because infants don't normally show physical signs of the disorder. So again, without detection from genetic screening, they may go undiagnosed or untreated, and then there could be permanent damage in the form of mental retardation. So some companies that offer this genetic testing are listed here. You can read them for yourself. There is some controversy associated with genetic testing, and that is the fact that newborn screenings are required by law without parental consent, and the Constitution protects citizens' property from invasion of government. The Fourth Amendment specifically, protects citizens from seizure of private property. There has been known that hospitals sell infants' DNA samples to research facilities, and several lawsuits have been filed due to unconsented use of infants' blood samples in scientific research due to that. So there is some controversy, but that concludes my presentation. Here are the references. I appreciate your time, and I wish everybody the best of luck in the course. Thanks.